Hi, my name is Agata. I am a PhD student at Silesian University of Technology in Gliwice, Poland. The poster I'd like to present today is about exploring mouse transcriptomic landscape. The mouse is a widely studied animal, as in many aspects its biology is conserved to ours, and it constitutes a valuable model organism. However, the complexity of its transcriptome is not yet fully elucidated. One of the mechanisms responsible for this is alternative splicing, which has been reported to be characteristic of almost all genes in mammals and may be one of the most widely exploited mechanisms responsible for increased transcriptomic and proteomic complexity. We present the results of studying the splicing events in data from an experiment focused on neuropathic pain in mice. In our study, we concentrated on finding commonalities in the collection of fairly diverse samples to expand the currently known mouse transcriptomic landscape. We had 3 batches of data in each different gene knockouts were induced in different structures. Each batch contained also a control group. For each type of sample, there were 4 replicates, resulting in 88 samples in total. We hypothesized that the inclusion of different pathological factors should allow detection of the spinal cord novel alternative splicing events characteristic under all conditions. For the purpose of this study, we used Splatter software, which builds an augmented splicing graph based on known annotation and then expands it with new events. Currently, it allows for detection of 6 different types of events, but we've decided to exclude intron retention because of our lab protocol. Splatter reports PSI value, which is the proportion of inclusion ratio versus inclusion plus exclusion ratio. That is, for example, percentage of events containing exon versus those who do not contain it. We ended up with 3 groups. One where both events were already known, another where both are new, and third one when one is new and one is old. For the first 2 groups, we've decided to set a threshold on PSI standard deviation and treat those which had standard deviation below 0.2 in all 4 replicates as valid. For the third group, we've also added threshold on the PSE value itself. It should be above 0.2. As you can see in table number 1, we found that some of the novel alternative splicing events are common among all samples in our study. The percentage of them is not very high, but we have to bear in mind that those are events detected in all 88 samples. If we look at the sample upset plot and Venn diagram below, we can see that the genes don't overlap between different groups. That made us wonder how novel events will change resulting gene ontology terms. You can see comparisons in the 2 panels in the right bottom corner, where the size of the dot is the number of genes and its color is related to p-value. In the lower one, we can see top 10 terms detected by events from group with both no events and how do they change when we add remaining 2 groups. We can see that it results in more genes being added to terms, among which many were related to nervous system. The upper one shows top 10 terms for each group and how do they look in other groups. We can see that when studying groups with both new events, the term response to axon injury becomes very significant. If we look at the terms detected separately for each group and event, on the sample plots in the right top corner of results pane, we can see a clear connection to the nervous system. Taking our study further, we've used BISBE and Interproscan software to see possible changes and implications on the protein level. The results can be seen in table number 2 and 3. Table number 3 shows selected events which were assigned to known proteins or protein domains, and we can once again see the connection to the nervous system. These results might indicate that the mouse reference model lacks information for brain tissue, but also reflect expected neuroplasticity. In the next steps, we would like to investigate selected novel alternative splicing events, as well as revisit the expression analysis with the improved mouse reference gene model. Thank you for your attention.